everyone. And this is Dr. Roy. I'm the coordinator of AATM Plasma Protein Therapy Working Group. And it's an honor and pleasure for me to be here and to be the host of this journal club. So I would like to request to our respected Secretary General, Dr. Ananda Gunashekhara sir, to give the opening remarks. Dr. Ananda sir, the floor is yours.
Thank you very much.
Thank you.
Good morning to Asia and good evening to Asia and good morning to Americas and the rest of the world. Thank you very much for joining, and thank you very much for those kind words, Dr. Kalyan. I'm very happy to be here today to give you these greetings. As we all know, AATM, or the Asian Association of Translational Medicine, is having more than 15 working groups right now. The plasma protein therapies group is one of them. All the groups are active. Unfortunately, the plasma protein therapy group was not very active until such time the new committee took over. Now it is on their top gear, doing so much of activities. And we have two wings in this particular group, plasma protein therapy. One side is for regulatory and establishment side. The other side is the academic. So this activity is an academic wings activity led by the co-chair, Dr. Sangeeta Pathak. She will be joined very soon, I'm sure. And the other wing is being done by Dr. our good friend Ranjit Ajmani. So also Ranjit, thank you very much for connecting us. The Plasma Protein Therapeutic Association, the leader in the globe, giving us the speakers, international speakers, Larissa and Sonia. Thank you very much, both of you, for joining and giving all this expertise to our group. So together with that, I would like to thank our group also, Dr. Sangeeta, for giving this leadership and making all these activities so beautiful for us. And also, I welcome all the members for this journal club. Dr. Kalyan is our active and good secretary. So he's going to do the moderation part today. Kalyan, now over to you. Thank you.
Thank you, sir. Thanks for the kind words. And now I'd like to request our invited guest speaker, Larissa Sarvenakova, and she is the medical director of Plasma Protein Therapeutics Association, and she will present on anti-SARS-CoV-2 antibodies in immunoglobulin G preparations and convalescent plasma guidelines in the United States. So, Dr. Larissa, over to you, ma'am.
Thank you very much for the kind invitation, and thank you very much for giving us this opportunity. So I try to put my slides on screen. I will share it with all. Can you see it?
Yes, pretty much.
Yes.
Okay, something is in the way. Let me just work on that. Okay, perfect. Good. Let me close this. Okay, so my name is Larissa Chervenyakova. I'm a medical director for the PPTA, and it is my pleasure to give you this presentation. Points of our discussion today will be, I will talk a little bit about PPTA, about plasma as a source for therapies, about human normal immune globulin as a therapy, COVID-19 and convalescent plasma, their uses, and about SARS-CoV-2 hyperimmune immune globulins, and SARS-CoV-2 specific antibodies in normal human immune globulin preparations. The Plasma Protein Therapeutics Association, or PPTA, is a dynamic trade association that represents a unique sector of the biologics and biotechnology industry. PPTA represents more than 1,000 human plasma collection centers in North America and Europe, as well as the manufacturers of life-saving plasma protein therapies. Our members produce approximately 80% of the plasma protein therapies in the US and 60% of those manufactured in Europe. Our members' manufacturing facilities are spread through the world. They are present in the United States, in Europe, in Japan, in Australia, as well as in China. PPTA works globally to advocate for access to and affordability of therapies for patients. It is engaged in constructive dialogue with regulatory agencies, and we collaborate with more than 20 patient advocacy organizations.
Larissa, can I just request you, Larissa, could I just request if you could just make it on a slide mode? Unfortunately, the slides are not moving. We are still on the first slide, Larissa.
It is very strange. It's moving on my screen.
Unfortunately, for most of us, if I'm correct, because I'm getting...
Make it slideshow first, then it will move.
Yes.
Now it's
moving.
Right now, ma'am.
Now it's moving?
Yes.
Yeah.
Okay.
Yes.
Okay, very good. So, yeah, okay.
Need to put a full slide mode, actually, Larissa.
Okay, so this was the second slide.
Yes.
This was the third slide, and now we are here with this.
Yes, Larissa, but still it is not in the slide mode. I can see your next slide as well alongside with it. So probably, you need to kind of, if you look at your screen, you need to make it in a complete slide mode. Yeah, please do this. No. Maybe no, this is a slider mode.
Yeah, I'm trying to figure out how to do it.
Sure.
Okay.
Maybe Larissa, no, it's a slider mode.
Any suggestions how I do it?
If you escape and then go on the screen, put it on a...
Yes, okay, next.
Okay.
Now start sharing your screen, and then on the right lower corner, you can find it in the full screen mode.
Maybe you can first share the screen, Larissa.
So share screen.
Right.
Let me try to close the other screen. I have two screens.
Sure. Yeah, if you now can go to the slider mode down.
Yes,
Yeah,
that's the one.
Yes,
That's the one.
Yes, please.
Can you see it now?
Perfect.
That's better.
This is perfect. This is perfect.
Better.
Yeah, it is.
Yes.
Yeah, now you can go to the third, fourth.
It's visible now, ma'am.
Now you can move forward.
Okay, very good. Oh my God. Okay, thank you.
Yes, perfect. Larissa, you are there. Thank you.
Okay, very good. Yeah, I had two screens, so because of that, it was discrepancies there. I'm very sorry.
No, it's absolutely fine, Larissa. Thank you.
Okay, PPT also administers standards and programs that help ensure the quality and safety of plasma collection and manufacturing and protect both donors and patients. We are also engaged in research with our member companies, mostly on donor health studies. So you all know these settings, and I probably don't have to spend too much time on differentiation between the plasma collected by apheresis and plasma collected plasma separated from the whole blood. So these are requirements for the collection of plasma here in the United States, and our companies collect plasma by apheresis. It is possible to collect plasma 104 times per year, twice per week, and this plasma is called source plasma. It is only used for the manufacturing purposes, to manufacture plasma-derived therapeutics. So the plasma collection is highly regulated process, and it is a very long process. Manufacturing of plasma-derived medicinal products is very lengthy. It takes approximately 7 to 12 months to produce therapies, in contrary to the small molecules production, which takes approximately 1 month to produce. So plasma contains more than 3,000 important relevant proteins, and only a small fraction of proteins is actually isolated from plasma and made into therapies. There are a few classes of therapies. They include immune globulins, coagulation factors, inhibitors, and albumin. The human normal immune globulin therapies are a unique class of biological manufactured from human blood plasma, and they are distinguished from monoclonal antibodies, which are produced from cell cultures by biotechnology processes. Clinical need for IG therapy increases each year due to better diagnosis, advanced therapeutic treatments, new indications, and awareness. Broad range of IG products is available on the market in the form of intravenous, subcutaneous, intramuscular immune globulins. The last one is not so often used anymore. Each IG brand has approved indications for specific medical conditions. So it takes approximately 1,200 plasma donations to treat for a year one patient for hemophilia, 900 donations to treat patients with alpha-1 deficiency, and 130 plasma donations to treat one patient with a primary immune deficiency. Plasma-derived medicinal products such as immune globulins and clotting factors, factor 9 and factor 8, are listed by the World Health Organization as essential medicines for adults and children. So it means that every country is supposed to have these medications for appropriate treatments. Human normal immune globulin therapy actually is used for two main purposes: to replace missing or malfunctioning antibodies, and to modulate the immune system in case of autoimmune and autoinflammatory conditions. These are approved indications for use of immune globulins in the United States. So let's talk about coronavirus outbreak, which happened more than 2 years ago. And United States government was very much involved in the beginning of pandemic in different activities. And one of them was to help with the development of therapeutics and At this point, FDA and HHS were involved, and huge efforts were made to help with the development of therapeutics and accelerate convalescent plasma collections through engagement in various collaborations with different agencies, academia, and blood and plasma collectors to produce sufficient doses of convalescent plasma for transfusion and hyperimmune globulin. As of April 23 of 2020, administration made comments during the White House Coronavirus Task Force briefing, encouraging persons to contact their local plasma donation centers to learn how they can help. The industry had a lot of different challenges, including donor recruiting and eligibility, need for simple, specific, and sensitive assays to select SARS-CoV-2 positive donors with high levels of antibodies, address all the logistics around shipments, produce sufficient volumes of hyperimmune globulin to the treatment of COVID-19 patients within a short time frame. A national campaign with a mission to encourage convalescent plasma donation from COVID-19 survivors to be used as a therapy in hyperimmune SARS-CoV immune globulin started on May 2020. This included coalition members which supposed to develop hyperimmune globulin, including COVID-19 Plasma Alliance, which was presented by different companies, main producers of immune globulin. So in the study, which is known as ITAC study, was multi-center study, international, randomized, double-blinded, placebo-controlled trial, which was done on 63 sites in 11 countries from October 8 to February 10, 2021. It included 593 participants. Half of them received hyperimmune globulin, half received placebo. So in addition to remdesivir, when not contraindicated, and other standard clinical care. It was randomized by the site pharmacy. The primary outcome was at day 7 by 7 category ordinal endpoint, assessment by pulmonary status and extra pulmonary complications ranged from no limiting symptoms to death. 579 patients were included in the modified intention to treat population analysis. Deaths and adverse events, including organ failure and serious infections used to define composite safety outcomes, were done at day 7 and 28. Pre-specified subgroup analysis was done for efficacy and safety outcomes by duration of symptoms, the presence of anti-spike neutralizing antibodies, and other baseline factors. Findings from this study were not positive for hyperimmune globulin. Compared with placebo, the group which received hyperimmune globulin did not have significantly greater odds of a more favorable outcome at day 7. Infusions were well tolerated. Also, infusion reactions were more common in the hyperimmune globulin group, 18.6% versus 9.5% for placebo. The percentage with the composite safety outcome at day 7 was similar for the hyperimmune globulin and placebo group. The odds ratio for the day 7 ordinal outcome did not vary for subgroups considered, but there was evidence of heterogeneity of the treatment effect for the day 7 composite safety outcome. Risk was greater for hyperimmune globulin compared with placebo for patients who were antibody positive at the day of the entry in the study. So here I just show you that immune globulins from 4 different companies were used, and there was no difference in regard to the outcome with the treatment. Conclusions from this study, as I said, were not favorable. This study was the first well-powered and for the treatment of hospitalized patients with COVID-19, when administered with standard of care, including remdesivir, SARS-CoV-2 hyperimmune globulin did not demonstrate efficacy among patients hospitalized with COVID-19 without end organ failure. There was no heterogeneity of treatment effect and efficacy among patients without endogenous antibodies compared with those with endogenous antibodies. But there was heterogeneity of treatment effect for the primary safety outcome. Risk was greater for hyperimmune globulin compared with placebo for patients with endogenous neutralizing anti-SARS-CoV-2 antibodies at the time of the treatment. It was very unfortunate, but the important thing was also, I still believe actually that not everything what was possible to do was done with hyperimmune globulins, and I hope that the studies will continue in the future. So we now go to the immune deficiencies patients who needed protection from infection at the time when pandemic started. And they were questions in the industry and within the patients community about whether pre-pandemic immune globulin therapies contain cross-reactive antibodies, which are present in plasma of donors to seasonal coronaviruses, which can also recognize SARS-CoV-2, and do current immune globulin therapies, which were available at the time and which are available now, contain SARS-CoV-2 specific antibodies. So a lot of studies were done, and they were kind of encouraging and discouraging. I just list them here for your review, and it was clear that pre-pandemic antibodies, pre-pandemic immune globulins, even so they contain cross-reactive antibodies with seasonal coronaviruses, do not react in neutralizing assays with SARS-CoV-2. So they cannot provide protection to the patients if they get infected with SARS-CoV-2 virus. But our companies collected plasma, and in these plasma pools, collected plasma pools, and then in final products, they started to check for the presence of antibodies. The difficulties were not only with the detection of antibodies, but also with the ability to do neutralization assays, because these assays cannot be performed outside of BSL-3 condition laboratory, and these laboratories are not easily available in the countries. So finally, in 2021, in February of 2021, the paper was published, which was done by one of our member companies. The testing of plasma pools were performed, as well as testing of IV batch products. And tests were, plasma was collected in Spain, in Europe, other European countries, as well as in the United States during different period of times. And in July and early September, it was possible to see that immune globulins started to show presence of antibodies against SARS-CoV-2. So a little bit later, plasmas collected in European countries, Germany, Czech Republic, and Slovakia also were positive for SARS-CoV-2. And batches of immune globulins tested made from the United States plasma were showing positivity in November, October of 2020. So this group actually continued to do study, and in January of this year, they provided data which showed increasing rising titers of antibodies in normal human immune globulin preparations. And what was important, neutralizing activity was showed also against wild-type virus, alpha, beta, gamma, and delta variants of concern. And this is the graph which shows increasing titers of antibodies in immune globulin preparations, the highest in the United States, Germany, other products. It is listed different products, but I cannot show it to you. You can go to the paper and look at that. But as you can see, in all the countries, the titers of antibodies were rising very nicely in normal human immune globulins, except for Slovakia. So this study presents data which were done by the academia. They took different immune globulin preparations with different expiration dates, and we're looking for the presence of antibodies within these immune globulins. And you can see this is the product which expires in December of this year, and this is a later product with expiration date in December of next year. And you can appreciate very much the increase in titers of antibodies in these different preparations of immune globulins through the year, through the different time period. Here it is a sample of convalescent plasma serum shown on the right. And you can see that the titers of immune globulin, titers of SARS-CoV-2 antibodies in normal human immune globulins are much higher than in a sample from the patient, convalescent patient. So, and this is just to show you again the differences and titers. It is for more recent preparations of immune globulins, and you can see that the titers increase over time. And this is the sample of immune globulin with expiration date of December 23. So all this is very nice, but it is still are the remaining questions. What are protective levels of SARS-CoV-2 specific antibodies in IgG? We don't know the answer to that. Would immune globulin made from plasma collected earlier during the pandemic provide protection from SARS-CoV-2 variants which have emerged more recently and can emerge later on? And could vaccination of general population and donors be sufficient to maintain protective levels of antibodies in IgG therapies? There are still remaining regulatory issues which are require standard for IgG with established antibody level and standard assay for detection of antibodies, protective antibodies with neutralizing activities. With this, I will finish, and thank you very much for your attention. I can take any questions you might have.
Thank you, Dr. Larissa, for your nice presentation. And I think the question and answer session will be later after the presentation of Dr. Sangeeta. So I would like to invite our co-chair, Dr. Sangeeta Pathak. She is also an associate director and HOD of Max Super Specialty Hospital. And she will present effectiveness of convalescent plasma therapy in Indian patients with COVID-19. So Dr. Sangeeta ma'am, over to you, ma'am. Ma'am, can you please unmute yourself? Dr. Sangeeta ma'am, we can't listen to you, ma'am.
Can you hear me?
Okay, yeah, loud and clear, ma'am.
Yeah. And are my slides visible?
Yeah.
Can you see my slides?
Yes, we
Yes,
can.
ma'am. In the full screen mode. Okay.
Thank you so much. Okay. Okay, thank you. So I'll be presenting about this study, convalescent plasma therapy in COVID-19. And this was done by our hospital and was published in this Blood Cells, Molecules and Diseases. And it was effectiveness on of convalescent plasma in Indian patients with COVID-19. And this was a group of our own hospitals, 3 hospitals which did this study, and the major hospital was the one that I work with. Now I can't. Okay. So the study design, the study design was that Max Hospital, they studied the potential use of convalescent plasma therapy in patients with COVID-19, and it was a multicentric retrospective observational case control study of total 1249 patients. But out of these 1249 patients, 170 were excluded due to the unavailability of sufficient clinical data. And the remaining 1079 patients were included in the final analysis. This study was conducted from 1st of May 2020 to 31st August 2020 in patients with moderate and severe or life-threatening COVID-19 who required the oxygen therapy. Then this was the study design that we used. So COVID-19 patients on oxygen admitted at the EHR, and this was between the 1st May 2020 to 31st August 2020. Then in this, we excluded 170 patients because of the unavailability of data. Then patients with complete data were 1079, as I already told you. Then patients admitted in the wards were 385. Out of these 385, 60 were on best supportive care plus the convalescent plasma therapy, and 325 patients were on best supportive care only minus the CP. Then patients admitted in the ICU was 694 patients, and out of that 333 patients, 333 patients were given best supportive care plus the CP, while 361 patients were given only the best supportive care. So this was the basic study design. Then what were the inclusion criteria? The eligible patients include the adults who were above 18 years with evidence of SARS-CoV-2 infections, which was confirmed by RT-PCR test of nasopharyngeal and oropharyngeal swab. And criteria for classifying patients into moderate and severe were as per the clinical management protocol, which were given by the Ministry of Health and Family Welfare here in India. So the moderate disease included pneumonia with no signs of severe disease and having one or more, that is dyspnea, fever and cough, SpO2 less than 94% on room air, respiratory rate was above 24 per minute. Then the inclusion criteria for the severe disease, this I talked about the moderate disease. Now for the severe disease, patient having one or more of the following conditions: clinical signs of pneumonia plus one of the following: respiratory rate more than 30, then ventilation or SpO2 less than 90% on room air, ARDS, new onset bilateral opacities and SpO2 or FiO2 less than 315, sepsis, septic shock, and patient meeting these criteria and requiring oxygen therapy were included in this study. Then donor selection criteria, we all know that. What were the donor selection criteria? These criteria were again and again changing with the time. We were experiencing a change in the donor selection criteria. But at that time when I did this study, the criteria were as follows: prior diagnosis of COVID-19 documented by a lab test. Then the second was complete resolutions of the symptoms at least 28 days prior to the donation. Then complete resolution of symptoms at least 14 days prior to donation. In such cases, if the patient has been discharged without a negative RT-PCR, 2 samples of naso and oropharyngeal swab have to be sent for RT-PCR for COVID-19, collected 24 hours apart, and negative result ascertained before considering the recovered patient as a potential donor. So just to tell you this, these criteria of selection kept on changing here and there again and again. And at that particular moment, these two, the second two in A and B were very important for us. Then the third one, only male and nulliparous female donors of weight more than 55 kg. We all know that even for the other plasma, we only take nulliparous females. Then donor with hemoglobin of more than 12.5 and platelet count of more than 1.5 lakh per microliter of blood and TLC within normal limits. HIV, HBV, HCV, serologically and NAT negative. Then syphilis, malaria negative donors and total serum protein more than 6 grams. And all donor selection criteria which were followed as per the Drugs and Cosmetics Act, which are there in India. We had to follow all those criteria as well. Then, in continuation with the donor selection criteria, in the absence of a standard viral neutralization test and limited possibility of assessment of neutralizing antibody titers of more than 160, 1:640, we adopted a policy of the use of an assay to assess the quantitative antibodies of anti-SARS-CoV-2 antibodies prior to harvest of CP from the eligible donors. And in our larger center, that is one of the center contributed the highest numbers to this, and in the larger center, the available platform which we used was an FDA approved, that is the Diasorin quantitative IgG antibody assay against the S1 and S2 protein of SARS-CoV-2 virus, and the cutoff of 15 AU per ml was taken for a positive result, which is equivalent to 1:40 plaque neutralization antibody, and 80 AU per ml is equivalent to 1:160 plaque neutralization antibody assay. Then in continuation with that, volume of donated plasma was 400 ml. The first 200 ml was infused over 1.5 hours, and if the patient did not show any improvement after 24 hours, based on the decision of the treating physician, another unit of plasma was then administered. What were the exclusion criteria? The exclusion criteria were patient not requiring oxygen therapy, obviously, were excluded, and pregnant and lactating mothers, they were also excluded. Now, these were the study approvals that were got. We all know that the institutional ethics committee clearance has to be there, and a proper informed consent for various treatments, including the convalescent plasma therapy use, was taken from every patient. Now, there were two objectives of the study, and the primary objective of the study was to study the 28-day mortality in patients who received convalescent plasma therapy versus those who did not. And the secondary objective was to assess if the CP reduced the chances of patients going on to invasive ventilation, and whether it offered any mortality benefits in the critically ill patients on ventilator and on vasopressors. Now, the statistical analysis. Baseline characteristics which were used were age, gender, comorbidities in the plasma, and no plasma group were compared with the chi-square test. The number of patients on invasive and non-invasive ventilators, mortality in plasma and no plasma group, and various subgroups of patients were also compared with the chi-square test. P-value less than 0.05 was considered statistically significant, and the sample size in two groups was adequate to detect the difference of at least 10% in the overall mortality between plasma and no plasma group, with power of 80%, when the mortality in ICU cases in the standard care group is 25%. I'll come to the results. The mean SD age of patients was 60.1 plus minus 12.1 years versus 58.9 plus minus 13.8 years in plasma versus no plasma group, with a P of 0.231. Then males were significantly higher in the plasma group, 80% versus 72.3%, with a p-value of 0.015, and less of younger age, that is 12.3% versus 19.2%, with a p-value of 0.023. There were more females in the plasma group with multiple comorbidities than in no plasma group, and specific comorbidities examined were hypertension, diabetes mellitus, coronary artery disease, and hypothyroidism. Now I'll talk about these 1079 patients which were on oxygen and which were taken in this group. The overall mortality in patients was 22.4% and 18.5% in the no plasma group. Total 694, 64.3 patients required transfer to ICU to the intensive care units. 333, 48.0% patients received convalescent plasma in addition to the best supportive care, and remaining 361, that is 52%, were on best supportive care only. In the ICU patients, mortality was 25.5 versus 33.2 in plasma versus no plasma group. P was 0.026. Reduced mortality in plasma group was particularly seen in patients of age group of 60 to 74 years, 26.7 versus 43%, with a p-value of 0.004, driven mostly by females of this age group, 23.1% versus 53.5% with a p-value of 0.013. Now, in the main hospitals with experience with the use of CP was maximum. Out of 342 patients in ICU, 239, that is 69.8%, received plasma, while 103, that is 30%, did not. Overall mortality in plasma group was 49 out of 239, 20.5%, as compared to 36 out of 103, that is 36.9 in no plasma group. Then out of the 694 patients with severe COVID-19 in ICU, 101 patients worsened and on non-invasive ventilation and needed to be shifted to invasive ventilators. 54, that is 53.46% of patients were given CP. There were 316, that is 45.5% patients who remained on NIV only, and out of these 157 received the CP. Overall, CP did not reduce the chances being put on invasive ventilation from NIV. And patients on ventilator had significantly lower mortality, the mortality in plasma arm, that is 37.2 versus 49.3. This was particularly, as I said, so far on patients on invasive ventilation with mortality of 63.9 versus 82.9% in plasma versus no plasma group. I'll show you this to understand better in a tabular form. This I'm just bringing the exact paper. I'll show you more better in a table form. You'll understand it more there. Patients on an invasive ventilator and on vasopressor had high, almost 90% mortality in both the groups. And 26 out of 333, that is 7.8, received second unit of CP as per the physician's decision. And 10 of these patients, that is the 38% of the patients, they died. So now as I told you this, I'll show you that in a study we found maximum benefit of CP in these patients. So the type of patient you can see on the left hand side, ICU patient 60 to 74%, and the mortality in the plasma group was 26.7%. And when you see in the non-plasma group, that was 43%. Then in patients with one comorbidity, it was 22.3 versus in the non-plasma group, it was 36.5. Again, a significant difference. Then patient on one ventilator. Again, the mortality in plasma arm was 37.2 versus 49.3 in the non-plasma group. And patient on invasive mechanical ventilation, again, or in the plasma group, it was 63.9 as compared to in the non-plasma group, which was 82.9. So you all will agree that this study showed a significant difference in the plasma and the non-plasma group. And this, the results are same results put in a more graphical form. Same thing, need of ventilation. And the discussion now, the current study was undertaken to evaluate the effectiveness of CP in patients with moderate and severe. Similar study involving moderately ill COVID patients by Abolgasmi et al. Showed that significantly higher number of patients who received CP were discharged from the hospital within 5 days. So early use of plasma definitely had an impact. And a similar study by Zia et al. Also showed that there was less likelihood of patients who received CP to be transferred to ICU as compared to those who did not receive CP. In this study, it was 2.4 versus 5.1%. However, this was not statistically significant. And the patient outcome in our study was very similar to the results which are reported by also Salzer et al., where they found significant mortality reduction in severe and life-threatening COVID-19 patients, that is 2.7 versus 8.9. And the overall conclusion of the study was the use of convalescent plasma was associated with reduced mortality in severe COVID-19 elderly patients with above 60 years of age, particularly in females, those with comorbidities, and especially those who require some form of ventilation. This beneficial effect was lost when the entire cohort of patients across varying severity of illness was compared. And plasma did not seem to offer any mortality benefit in patients of moderate severity or those who were terminally ill. Further research into good mechanisms of CP in COVID-19 may help predict the good responders. So this result was in August 21, and we have traveled a lot after that, and this study that time concluded in this manner, and now we have better results with the CP. So thank you so much for your patient listening. Over to you, Kalyan.
Thank you, ma'am. Thank you for your very nice presentation. And as we are running out of time, I think we have very little time for a question and answer session. So if anyone have any questions, please go ahead.
If you are interested, I can show you one slide because I looked at publications related to the use of convalescent plasma and then to clinical trials which are ongoing or have results or whatever. So if you allow me to do that, I can share it with you. I try my best this time.
Please do that.
Sure, ma'am. With pleasure.
We'll be more than happy to see that, yes.
Okay, good. Can you see it? No.
Not yet. You have to share screen first, Larissa.
Yeah. Just a second. Yeah, I'm trying to figure out how to do it
Button
again.
is there in the lower side of your screen.
Yeah, I
Press
know.
that share screen. Yeah.
Share screen, and then no, it shows me again not what I want. Okay.
We can see your screen now. Yes.
You can see my screen now?
We can see your entire screen. Yes. This is your presentation, I suppose.
Yeah, just a second. I need to use different folder. Okay, good. I thought that you will be, can you see?
Yeah,
you
we
see
can
my-
see. Yes.
Okay, this is what I wanted to share with you. If you look at this slide, actually, this is the number of publications in PubMed, which goes to, I try to make it, okay,
My
can
God,
you see?
huge number.
Okay, so this is the number of publications in 2020, 21, and 22. And this is the number of clinical trials on clinicaltrials.gov, 201 trials, 11 which are not recruiting yet, 48 recruiting, 16 active not recruiting, 16 terminated, 4 of them have results, and 12 completed from the US, Brazil, and Argentina. Actually, it is 13, 5 withdrawn, and 13 have unknown status. 164 of them are interventional, 27 observational, 1 is related to registry, and it is funding is NIH funded 7 of them. Other US agencies provided funding for 6, industry for 14, and others 176. So I think that it is like we will expect to hear more about use of convalescent plasma. Interesting that people are doing a lot, but they are not really publishing because majority of studies published is coming from the United States. It could be because different countries may have started later on to do research in this regard. So now I will get out of the screen, but I thought that it is really interesting to see this data and kind of look forward to what to expect.
That was very interesting. Huge
Yeah.
number of publications.
So your publication among them, one of them.
Correct.
Actually, it was 2 publications, I think, from India when I
Yeah.
looked at the data.
Okay.
So
Over to
Larissa,
you, Kalyan.
can I ask you a question regarding a convalescent plasma?
Yes.
So can the donors who have just recovered from COVID-19, can they donate plasma for fractionation?
Yes, it is. Yes, they can donate, but I think it is 10 days waiting period after the recovery. And it doesn't matter. I think they don't have to be PCR negative. And also it doesn't matter. There is no regulation in terms of vaccination status. For donors of convalescent plasma for direct transfusion, they are not allowed to donate if they were vaccinated.
Okay.
So
you.
it is because still it is not clear how, you know, how it will actually affect the recipients of the convalescent plasma. So the regulation right now, the last regulation was issued in January of this year from FDA. And
Thank you.
as well, it is changing all the time, as Dr. Patak mentioned. It is the same everywhere, depending on how much data we are getting at the time the regulations may change. It is not final.
Yeah.
And it is still used under emergency use authorization.
Correct. Correct.
Yeah.
And only one site, I believe that only Mayo Clinic is licensed for doing the studies. Other studies are done under IND and so on. But it is like speeded process for that.
Yeah.
The biggest problem, I believe, is detection of the antibodies, because everybody is using different methods for detection of levels of antibodies in plasma. And sometimes the ELISA which is used is actually are not telling us the whole story.
Okay, so Larissa, what about the people who have just got their third jabs or the booster dose of COVID vaccine? Can they donate for fractionation?
Yes.
Okay. So do we have to hold it for 10 days or not?
It is a good question. I don't recall, but probably yes. Maybe 1 week even.
Okay.
I didn't. Mm-hmm.
In Dr. Kalyan, in India, we have a policy of not donating plasma after till 14 days of the jab. So after 14 days only they can donate.
Ma'am, would you follow it here in Bangladesh as well?
Yeah, we follow that.
Yeah, WHO. You follow WHO or you follow WHO? Yeah.
Okay, thanks a lot.
It is interesting that it is different in every country.
Yeah.
Yeah.
Science is the same, but there are differences in regulation.
So I'll tell you one interesting thing, that in India, it was 28 days earlier. When we started in the early times, it was that you cannot donate till 28 days you get a vaccine or this. Then we put up a letter to the Ministry of Health and Family Welfare that this is not, it is scientifically not required. And they agreed. And they came down to 14 days.
Yeah. So in India, it is Ministry of Health which regulates. Yes, it is. You don't have the...
Though we have a different cell, drugs and it comes under Drugs and Cosmetics Act, but yes, the Ministry of Health and Family Welfare does take decisions also in such cases. And this was a pandemic, so they had a special power also to, means that particular cell was looking after, and we had addressed our problem to them, and they did take a decision, and it was a decision which was put on, means taken on a good time, where we needed donors and we needed help from them, and we welcome that decision, of course.
Yeah. Well, unfortunately, we expected more, yes, from convalescent plasma and from hyperimmune globulins, and it is still debated, yes, how effective it is. It is really effective for very small population of people, but it is probably still worth of pursuing these endeavors. Especially, and I didn't see any study which will test the use of hyperimmune globulins for patients with immune deficiencies, but there are few publications when convalescent plasma was used to treat patients with immune deficiencies who were infected with COVID, and they did, at least maybe only positive publications are coming out, but at least there are few indicating that for these groups of patients, it may work very well.
Yeah. Over to you, Kalyan.
Thank you, ma'am. Thank you, Dr. Larissa, and thank you, Sangeeta, ma'am, for your mesmerizing presentation. And it is the time for vote of thanks, and I would like to invite our chair, Dr. Ananda Gunashekhar sir to give the vote of thanks. Sir, over to you.
Thank you, Kalyan. Thank you very much, everybody. It was a wonderful journal club. We learned a lot. Thanks, our international speakers, Larissa and Sangeeta. Thank you very much for your presentations. And thank you very much for moderating so effectively, Kalyan, as usual. My thanks goes to you. Once again, I thank Ranjit for connecting us with the international community in plasma protein therapy. Ranjit, my thanks goes to you. And thank you very much for joining us from several countries, from Europe, from America, from Asia. And it's wonderful. This is the first journal club of our group. And it's a brainchild of our co-chair, Sangeeta. So Sangeeta, we need more and more from you activities in order to make this group and the AATM success. So in absence of my president, he's busy today with the Director General in India. Actually, for your information, he's one of the agenda point for today's meeting with the Indian Director General is giving the support for Sri Lanka in blood transfusion service, during this crisis period. So he's connecting today with the Director General of Sri Lanka today. And in our previous meeting, we hope to join the Indian the Health General also in order to support for Sri Lanka. That's an extra news. And I'm compelled to do that, giving my heartfelt gratitude toward the international community, those who are supporting us at this crisis time. But anyway, so it's all and all good. Let's do more programs. Let's do more of activities like this. And over to you, Kalyan.
Thank you, sir. Thank you very much. And I thank you all for joining, and especially Dr. Sangeeta, Dr. Sonia, and Dr. Larissa. Heartfelt gratitude to you. Thanks a lot, and hope to see you soon. Thank you very much.
Thank you all.
Thank you.
Bye.